Hello everyone, and welcome to this audio podcast. This audio podcast today is part of the Quail N Crux. This audio podcast is part of the Ricochet and Ruckus. This audio podcast is part of the Bedlam Labostics. This audio podcast is part of the Shitstorm Squadron, and this audio podcast as part of a ZtormLabostix production. Through this audio podcast today, looking at some aspects in around Huntington's and genetics. This audio podcast sort of follows in from some of the molecular bases of Huntington's disease from the previous audio podcasts in this series. So in around Huntington's genetics, the number of the amino acids cytosine, adenine, and adenosine, and guanine is where the repeats determine the clinical manifestation. That is to say, that depending on the number of repeats, as sort of mentioned in the previous audio podcast around the molecular basis, that there are different sorts of, depending on the number of repeats of particular of the particular amino acids in amongst the DNA. There are then differences or variations in the clinical manifestation of Huntington's. So a strong inverse correlation exists between the cytosine, adenosine, and guanine repeat lengths and the age of onset, as for example juvenile, which is Huntington's onset before an individual reaches the age of 20 years of age or 20 years old, is often associated with over 60 sort of amino acid, those particular amino acid repeats. The phenomenon of anticipation, where onset occurs at a younger age, is also successive at a younger age in successive generations, is also frequently linked to paternal transmission due to greater repeat expansion in the male germline, in amongst the male germline. There have been some published data in around the mid 90 in around 1994, where investigators had informed that the amino acid repeats in around cytosine, adenosine, and guanine repeat instability and their sort of impact on the impact had shown that Huntington's instability, Huntington's disease instability is sort of occurred in around different differences or variations of transmission. According to the investigation, around 45% instability during paternal transmission, and amino acid repeat length had increased, and 29% of instability had been sort of evidenced or indicated in maternal transmission, with smaller changes or even decreased lengths of the amino acids of the amino acids which had been observed in particular investigations. Huntington's is also transmitted in an autosomal dominant fashion, meaning meaning an individual with an affected parent has around 50% chance of inheriting the sort of genetics. Genetic factors also play a major role in the condition in Huntington's, accounting for about 60% of variability in age of onset, and with the rest of influenced by the environmental or other genetic modifications. An inverse relationship also exists between amino acid repeat lengths and age of onset, although repeat length explains only around 60 or 70% of the variability. The remaining influenced by the remaining sort of aspects of genetics is also influenced by sort of genetic modifiers such as the genes involved in DNA repair, which can also alter somatic instability and promote further expansion of the alleles and the amino acid sort of line and the amino acids and the amino acids. And according to some data, historically, the largest number of amino acid repeats in cytosine, adenosine, and guanine repeats have been evidenced and recorded as in around 250 repeats. That draws this audio podcast to a close. This audio podcast as part of the Quail N Crux. This audio podcast as part of the Bedlam and Labostix. This audio podcast as part of the Ricochet and Ruckus. This audio podcast as part of the Shitstorm Squadron. And this audio podcast as part of a ZtormLabostix production.